Hello and welcome to an introduction to the HE Toolkit R package. This is the first of two training workshops which provide users with instructions in the use of the Environment Agency's new Hydro-Ecology Toolkit, which is an open source R package for assembling, processing and analysing hydroecological data. My name's Andy Davey. I'm a principal data scientist in the Hydro-Ecology team at APEM. I'm a freshwater ecologist by training, and I've worked in water resources, water quality and catchment management for around 20 years. Since 2021, I've been very closely involved in the development of the HE Toolkit, leading a series of projects and working very closely with my co-presenter today, Mike Dunbar, who is the former team leader of the Environment Agency's National Hydroecology team. And it was Mike's pioneering research which led to the development of the HE Toolkit, and Mike was responsible for commissioning and designing the toolkit. I mentioned we've split the training for the toolkit into two separate sessions. This first recording, workshop 1, provides a general introduction to the HE Toolkit. It's very much aimed at anybody working in the freshwater environment who is interested in being able to access, process and analyse open source environmental data sets. Workshop 2, which will be covered by a separate recording, builds on Workshop 1 and is aimed at more advanced users, data analysts who are interested in developing and applying statistical techniques to understand and predict ecological responses to changes in flow. So in this first workshop, we're going to start off by providing a bit of general background about what hydroecological models are and how they can be used to aid decisions around water resources planning. We can then look in a little bit as an overview really about what the HE Toolkit is and what it does. And then we're going to be working through the sequence of sort of tasks or workflows, starting off with data import, so looking at how we can access open source data via APIs, looking at how we can then process both hydrological and ecological sampling data, focusing in particular on macro invertebrates and river flows. And then we'll look at how we can bring that data together and use it to produce hydroecological validation plots, which allow us to eyeball the data to determine whether or not there are important influences of flow on the ecology. This training workshop is accompanied by a series of practical exercises which we have hosted as an RStudio project on POSIT Cloud. Details of how to access these additional training resources will be posted alongside this video. The key thing to emphasise, I think, is that today's workshop is very much a foundation and introduction to the HE Toolkit and how it can be used. We're not going to go into too much of the technical details of behind some of the functions within the toolkit, but really focus on the broad benefits of using the toolkit. I'm now going to hand you over to Mike to give you a little bit more background about some of the research studies and policy developments that paved the way for the development of the HE Toolkit.
I could talk for hours on this, so I'm going to give you a brief summary. I'll talk about three aspects to this, with one slide for each. The Hydro-Ecology Toolkit is not all about macroinvertebrates. Much of the history does come from that angle. The key initial moment was work undertaken by colleagues in the late 1990s in what's now Lincolnshire and Northamptonshire area, which led to the publication of the paper describing the macroinvertebrate life score. This started the practice of graphing time series of life alongside a hydrograph, which then developed into the agency's HEV or hydroecological validation procedure, including other invertebrate metrics as well. This has been a mainstay of our work over the past 20 years, but it does have its downsides. For example, it works best with long runs of monitoring data, and it's a description of the historical situation. It's not able to generalise or predict. We began in 1999 to improve the statistical basis for the association of life and flow. But things really took off in 2005 when we started using mixed effects models, which we'll cover in the second workshop, to discover the relationship across multiple sites and to begin to explain why we might be seeing the differences that we saw. A key result was to bring together data from River Habitat Survey, that is, data on physical modification of the channel and banks, especially the extent of channel resectioning, the wholesale widening, straightening and reprofiling of the channel for flood management and land drainage purposes. We showed across multiple data sets that the invertebrate communities in more engineered channels declined more strongly during periods of low flow, and conversely, in less modified channels, they were more resilient. As we noted in the journal papers that we published, this has implications for water resources and drought management. At that stage, the data assembly process was still quite onerous and time-consuming. We continued to develop and publicise the approach over the next decade and apply it to various questions in water resources, but there was limited uptake outside of the Environment Agency. One reason for this was the quite involved sequence of data access and processing tasks required to bring the various data sets together. Quite frankly, the whole process was a pain and was crying out for some standardization. The third angle of the background comes from the evolution of the environmental flow indicator and our clarification of the role that can be played by local knowledge and data to formulate an alternative flow constraint. This is encapsulated in the EFI position statement, where the Environment Agency notes that we need to be at least as confident in the local flow constraint as we would be in the EFI. The position statement covers several situations, including the impact of existing licensed abstractions, risk of deterioration where there is headroom in licenses, and applications for new licenses. The position statement includes an annex with some ideas on supporting data and interpretation. Interpretation needs to be supported by statistical analysis, and while there will undoubtedly be some situations where data will need to be interpreted on a site-by-site basis, in many cases, particularly upstream of the lower reaches of rivers, where we generally don't have sufficient data for the sites we're interested in, and where there's benefit in looking across time series data from multiple comparable sites along a gradient of pressure from water abstraction and potentially other pressures too.
Thank you very much for that, Mike. Now, before we talk about any of the technical details of the HE Toolkit, I thought it'd be useful to give you a bit of an overview really about what the HE Toolkit is and what it does. As Mike alluded to, the HE Toolkit is really a consolidation of the existing R code that's been developed by Mike and his colleagues at the Environment Agency, and also by ourselves at APEM through various Environment Agency projects. That code has been developed to help assemble, process, visualize and model hydro-ecological data. But the problem was that code was spread across various different projects. So what we wanted to do was bring it all together, wrap that code up into a series of discrete functions, and then package all of those up into a complete R package. So basically that brings all the code together, and by representing its functions makes that code much easier to apply to your particular data sets. The HE Toolkit package is freely available to download and install from the GitHub page. The link is there on the slide if you want to explore our GitHub pages. And it's really been developed for use by hydroecologists, irrespective of whether you're in a water company, you work for a regulator, you're a consultant, or you work in the academic community. If you're working with hydroecological data or any kind of open source environmental data for that matter, the HE Toolkit provides functions which really help to sort of speed up and automate the process of working with that data. I think it's helpful to think of the HE Toolkit as a set of building blocks, if you like. It's not an all singing, all dancing tool where you can just input some data at the beginning, press a couple of buttons and then get an all singing, all dancing predictive model out of the back end. It's more a set of tools, really, that we can use and link together to, say, speed up that process. So rather than having to write lots of code from scratch, a lot of these functions do a lot of the legwork for us and speed up the process. Also, by using the HE Toolkit and releasing it out there into the hydroecological community, We hope it will also tend slightly sort of standardise, if you like, the way in which we develop and apply these kinds of predictive models, and that over time that will provide a stronger evidence base to give us insights into how river flows influence the ecological status. It is a toolbox, to say, as I said, rather than an all singing or dancing model. So you will still need to write your own R code to some extent to link together the component functions. We're going to cover some of that in this workshop and also in the companion workshop that deals with the statistical modelling sides of the toolkit in a bit more detail. Plus, there's examples of how you can apply the toolkit as an entire workflow in the vignette on our GitHub page. Being an Environment Agency developed tool, it is a little bit England-centric. So it is set up primarily to work on data for English rivers. That may change in the future, and if you, the users, say that it would be useful to be able to incorporate data from other nations around the UK, for example, that's something that we could certainly look at. In the examples we're going to look at in the workshops, we do emphasise or focus quite a lot on benthic macroinvertebrates, simply because that is a biological receptor for which there's quite a lot of data, some very well-established community-level indices, and there's also quite a rich literature out there as well, demonstrating how these invertebrate indices respond to antecedent flow conditions. However, it's important to emphasise that the principles underpinning the HE toolkit, as well as the functions themselves, are applicable to any biological receptor. So you can use many of the functions that we're going to cover in these workshops to also analyse macro phyte data, algal data, fish data as well. The current latest working version of the HE toolkit is version 2.1, which we released at the end of March 2023. This incorporates various small enhancements and bug fixes, as well as a new function for importing water quality data. The HE toolkit is still evolving, however, and we would love to hear from you if you've got any further ideas or suggestions about how we could use to help shape the future development of the HE toolkit. There's more details at the end of this presentation about how you can get in touch with us with any suggestions. To install and run the HE toolkit package, you can install it directly from the GitHub page using the remotes package. The code on screen here, those 4 lines of code, are all you need to do to download, install and load the package ready for use. When you install the package for the first time, we'll also install any missing package dependencies. That is, any packages that the HE toolkit itself relies on to do the various different functions. When you use the HE toolkit on your own system, we recommend that you download and install it into an empty RStudio project. And also that you use the renv package, which is a package dependency manager. This essentially takes a snapshot of all the different packages, including the HE toolkit, that you've used to undertake an analysis. This means that you have a complete record of everything that you need to reproduce your analysis in the future, even if the HE toolkit or other packages are updated. Now, there's quite a lot of different functions, if you like, or moving parts to the HE toolkit. So just to map things out for you a little bit, we've put together this schematic. On the left-hand side here, we have the blue boxes, which represent all the different input data sets that we can bring in and analyse using the HE toolkit. So from top to bottom, we've got water quality data, river habitat survey data, environmental-based data about biological sampling locations, the actual biological or macroinvertebrate data itself, and then various different types of flow data, whether it's from the Hydro-Ecology Data Explorer, the National River Flow Archive, or other local or modelled files. We also use something called a metadata file, which is a starting point, which tells the HE toolkit the list of sampling sites or gauging stations that we want to extract data for. The flowchart then sort of breaks down the process into a series of steps. After the input data, we've got a set of functions for importing data, then some functions for processing the data, exploring or visualizing it, and ultimately for modeling the data, the end goal being to develop a predictive hydroecological model. The green boxes each represent one of the functions in the HE toolkit. So you can see there's a set of functions there for importing data. Those flow through to give us data sets which we can then process, visualize and model. The grey boxes are specific steps in a typical workflow where there isn't an existing function. Instead, we use a small amount of manual coding just to get those different data sets to link together. And examples of that code will be provided in the workshops as we go through. In this first workshop, we're not going to try and cover everything. The aim of this first workshop really is to show you how you can import and process and visualize the data and get it to a point where you can do an initial sort of visual assessment of any hydroecological relationships. The more detailed and technical aspects of how we then develop statistical models which formally describe hydroecological relationships, that's going to be covered in workshop 2. So on the slide here, you can see this slightly more restricted set of functions that we're going to focus on in this first workshop. And the ultimate endpoint for this first training session is to get us to a point where we can produce a hydroecological validation plot as shown on screen. This takes a set of processed hydrological data and processed macroinvertebrate data and plots those up as a time series so we can explore and investigate how changes in flow over time have impacted upon our biological metrics. The remainder of this workshop is split into a series of tutorials where we look at how we can use different functions from the HE toolkit to import, process and analyse hydroecological data. However, to do more than just describe how the HE toolkit works, we've also set up a series of practical exercises which give you, the user, an opportunity to get some hands-on experience of using the HE toolkit and also some example code that you can use as a template for developing your own workflows. To access these practicals, we've developed or set up an RStudio project which is hosted in the cloud, which you can access via the link which accompanies this video. To give you a quick tour of that online project, I'm just going to give you a quick walkthrough now. So accessing the link takes you through to the Posit Cloud page. Posit Cloud is just the new name for RStudio, and this is a cloud-based version of the familiar RStudio environment. You will need to register for a free Posit Cloud account. Logging into your account will then take you through to the practical workshop. You'll notice that will just take a few minutes for all of that to upload. And when it does, you'll see basically on screen in your web browser the familiar RStudio development environment. To accompany each workshop, we've created a set of example data sets and also some example R scripts, which illustrate all of the different functions that we're covering in these workshops. Here on screen now, you can see we've got the console over here on the left. We've got the environment listing the various objects that we create on the right. We've got a files list as well down here in the bottom right-hand corner. Once you've successfully logged in to access the project, the first thing to do is just to save a permanent copy. It simply clones a copy of the RStudio project over to your own personal workspace, which means you can work on it, change it, and save your edits. Again, that will just take a few seconds to clone across. There we go. And now we can just start to explore some of the files which we've made available for you here. The data folder contains all of the example data sets that we have created for use in these practicals. So if you want to, you can zoom into there. This particular example, we've got a CSV file containing some flow data. We've got some previously saved invertebrate data, and we've also got a metadata file, which lists out the biology and flow sites that we're going to analyse. We've got an R script as well, which provides some example code you can use if you were to install the HE toolkit from scratch in your own new RStudio project. For the purpose of these practical exercises, however, the HE toolkit, and all its dependent packages, and any other packages that we've used in these practical exercises have already been installed in the project here. So there's no need for you to download those from scratch. Instead, you can just navigate directly to one of the R scripts which contains the code for each of the practical exercises. So for example, we can click on practical exercise 1 and that loads the script for us in the top left-hand corner here. All we need to do to start analysing our data is to load the HE toolkit using the library command. So I can click onto row 6 and use the run button there. Then we can step through the script a line at a time or a section at a time and run the code. Outputs will then be returned in the console in the bottom left, and any plots will appear under the Plots tab on the right. Scripts are structured into a series of sections. So this first practical exercise, for example, first of all allows us to load the HE toolkit, read in some metadata, and do some initial setting up to provide a list of all of the site IDs. The second part of the script then imports some flow data. The next part of the script imports the environmental data. The next section imports macroinvertebrate data. We then import RHS data. Together, all of these sections can be linked together, so you can run the script either a line at a time, or if you want to, you can select multiple lines and run those together. You will see here, for example, that some of the tables of data that the script uses will pop up at the top here, so you can inspect those. It looks rather like an Excel spreadsheet. And any objects that you create, any data frames or model objects, will be listed out here in your environment. If you want to inspect an object that you created, a data table, for example, then you can click on the table icon on the right here, and again that will bring up a copy of that data table for you to inspect visually. Feel free to open up, run, and if you're feeling ambitious, please feel free to sort of tinker with and edit any of those scripts. It gives you an opportunity to see a typical workflow, and hopefully this will provide you with a useful template for doing your own analyses.